All right, I'm going to give you some notes on chromosomes, just so you have some general ideas that goes beyond a little bit of the karyotypes and other things you've learned about in the past about chromosomes. Um, so cytogenetics is basically the study of chromosomes. So what's in a chromosome? Mostly DNA and protein, about 50% of each. For a long time, I thought protein was genetic material because there's so much in the chromosomes. They're described by size and shape. Our number one chromosome's the biggest one, number two's the next biggest one, and so on. At the ends here, we've got the telomeres. We've talked about those already. They're basically a repeated sequence of DNA. Also, we got the centromere in the center, so that's basically a fraction of DNA here and here that match each other, that create this constriction that holds two chromatids together to make the chromosome. All right, went over that. In case you want to know what the sequence of the telomere is, it's TTAGGG, repeated over and over. Something else, sometimes you'll see these satellites on chromosomes. Looks like they got these little dealy boppers on the ends for those smaller chromosomes. Not really concerned about that. Karyotypes, we're doing a karyotype activity tomorrow, so don't worry about that. Most of our chromosomes are either metacentric, in which the centromere is in the center, submetacentric, a little bit off-center, or near the top, that's acrocentric. We don't have any of these telocentric chromosomes. Also, if it's not metacentric, it'll have a short arm and a long arm. The long arms are always referred to as the Q arms, and the short arms are known as the P arms. They'll talk about how you get karyotypes. We can get them a lot of different ways. Amniocentesis is generally the most common form. We generally only do karyotypes at baby for the most part. You can do it through chorionic villi sampling and fetal cell sorting, but that one's pretty expensive. But very limited miscarriage issues or things can happen with fetal cell sorting. And then they basically are going to squash it in the metaphase stage and then stain them so that you can see the banding patterns of chromosomes. All right. There are some chromosomal shorthands you might see if you do more research in chromosomes. Basically, it works. First number is how many chromosomes you have. The next, you either have XYs, that's your sex chromosomes. And then if there's any issues, they'll be listed after that. So if you're 46, got normal complement of chromosomes, XY, you're male. 46, XX, normal female. And then there's a few. There's Turner syndrome is one where you can be missing a chromosome and still survive. I'll go into that in a minute. Anyway, you can identify a gene through chromosomal shorthand as well. We won't go into that detail at this point in time. There, if you want to look at it. All right. Polyploidy can cause issues if you've got too many chromosomes. A lot of spontaneous abortions are due to being polyploidy, having too many where the meiosis didn't occur correctly. So two sperm try to fertilize an egg, or the egg was diploid, it didn't go through finish meiosis. So that will lead to death. That won't survive if it has three of everything. However, we can survive if we just got one of something. That's called aneuploidy, which is caused by non-disjunction. Basically, if pulled, one cell is going to end up with an extra chromosome. The other cell is going to be missing one of those chromosomes. And slides on how that happens. We do have a few trisomies and monosomies that are survivable. Trisomy 21 is one you should be aware of. That's Down syndrome. It's the most common. This one's the most survivable. Turns out maternal age seems to be a risk factor for all of these trisomies. They do have some medical problems, but generally treatable. Their developmental disabilities can be vary greatly. Some of them are very, very low people, and some of them do go to college, so they're not too bad off. Uh, Here's the maternal age and causing. Yeah, as you get older, it usually happens. You can see that it rises dramatically as you get older. People say, oh, after 40, you should really worry about that. But if we look at 40, it's roughly 1 out of 100 times you'll have it. That means 99% of the time would be fine. You have a child at 45, 1 out of 50, that means you got 49 chance. Well, I guess that'd be a 98% chance that you'll have a normal child. Um, you shouldn't have kids when you're older anyway. Trust me, I'm a father. I would not want kids. I'm in my 50s now, and I would not want kids now. All right, the various trisomies that are survivable. Generally, they're smaller chromosomes. Trisomy 18. We talked about trisomy 21, Down syndrome. Trisomy 18 is known as Edwards syndrome. These children have very serious medical and physical disabilities. They have a peculiar hand grasp that looks like that. Generally, um, these children don't live too long. They probably will not see age 5. Trisomy 13 is even worse, known as Patau syndrome. Generally do not survive past 6 months. Their eyes actually fuse together. This is probably where we get the legend of the Cyclops from, is trisomy 13. Lots of sex chromosome problems. Most seem to be survivable. Turner syndrome, where actually this is the only one you can survive if you're missing a chromosome. So you're missing one of the X's. You only are missing a sex chromosome. So, of course, all these people are female. Phenotypes generally short stature. They get this webbing on the back of the neck. They don't go through complete sexual development. They're not fertile. Some hearing impairment. They can have some issues. You get mosaic still because females still want to turn off an X chromosome, so it might turn off that X even though it's the only one it has. Triple X syndrome. 1 out of 1,000 females get this. Tall, menstrual irregularities, slight impact on intelligence, nothing too serious. You can actually look at their cells and see 2 bar bodies. That means instead of turning off 1 X chromosome, you turned off 2 to kind of normalize things. So it's really not too big of a deal there. Klinefelters, again, and not one. These people are generally, they don't know until they have a fertility problem because these boys will not be fertile. They basically have 2 X chromosomes. And sometimes they even get a bar body. So one of those X's will be turned off because you know your body knows you have too many. But some of your things don't develop correctly. Again, it's 1 out of 1,000 male births, so it's there, it's out there. XXYY, so probably had an irregular sperm or an unusual oocyte. Something went wrong there. Anyway, they're AAD, OCD, some learning disabilities, infertile. They will treat it with testosterone. XYY, sometimes known as Superman syndrome. 1 out of 1,000 male births, you got an extra Y chromosome. Normally, you're 96% normal, really, though you'll be a little taller, generally have more acne problems, some reading disabilities. There were originally studies that suggested that people with this were more aggressive. That's because they were only doing these karyotypes from people of prison populations. So they saw it more there, but when they actually looked in the regular human population, it turned out that it was about the same percentage. So it's not a big deal in terms of aggressive behavior. There's some other abnormal structural abnormalities. Things get deleted and stuff. We're not going to go too much to that. Sometimes a duplication like this happen. More genes, and the more extra things you have, more problems. Sometimes piece of chromosome will move from one chromosome to another, as in Robertsonian translocation, 5% of Down syndrome. So basically, an extra piece of chromosome 21 is moved to chromosome 14. They'll just have 2 21s, but then they got that extra 21 over on a 14. And inversions, these are just basically a flipping of the chromosome, which causes the genes to be backwards. It basically occurs something like this. The chromosomes will flip and cross over and then trade off the genes in a wrong direction. And the only difference between paracentric and paracentric is where the centromere is involved. Isochromosomes, basically the chromosomes cut in the wrong direction. So instead of going north and south in cutting, it goes east and west. So you end up with this weird chromosome that has the same genes on both sides. Sometimes there's ring chromosomes. The ends, basically the telomeres, are lost and create a sticky end, so they'll combine and form a ring. And basically that's all I wanted to cover about chromosomes. You'll do a karyotype activity tomorrow. Just to show that you watched this video, I would like you to send me an email. And in that email, I want you to type in the subject, I watched this. That's all I need. Send me that email, and I know you watched it, and I'll give you credit for that. All right, that's it. Goodbye.